Now we are. All right. Well, I didn't actually finish it. I was like 9 hours later. So
It's great.
yeah.
It's a great deal.
That's what I keep hearing. I gotta admit.
So funny.
All
I
right.
don't know if girls are like as much as guys.
Let's lay out our schedule for the next little bit. And we're going to do eukaryotic gene regulation. We're going to do that again on Friday. And then, I think next week, in celebration of E-Days, we're going to have
No, class on Wednesday.
small group presentations on Monday and Wednesday.
I was the opposite.
You don't have to, you get to. Do small group presentations and earn points towards a good grade. Did your gel work out?
Like half of it doesn't have enough video in it, so like mine, I can't see it maybe very good. The other half looks really good though.
Okay.
So my half is good? Yeah. Hey, you couldn't tell the top two, especially yours were kind of.
Who else has run a gel recently? Did you run it with your PCR product?
I don't know if it worked or not.
And can you tell the class how that worked? The gel you ran?
It looks splendid.
Splendid.
It's just kind of funny because I ran 16 gels, or 16 wells, and I did them half size on one. So it was
So you ran one gel, and instead of having wells at the top, you had wells also in the middle, so you're only running half as long.
yeah, so half as long. And it was funny because I did it 1.1, 1.2, so and then 2.1, 2.2. And then if you took a picture of it, it looked like there was 2 Fs. I don't know. I thought it was funny. The ones that worked. I don't know. Okay, it works.
So,
You're gonna be fine.
so this is your gene, or this is back?
The back and my gene. It's
Both back and your gene work. What is your gene?
106
TMEM 106C.
C.
Okay. So that is, so we've been able, finally someone's been able to get a gene other than back to work. And did you test old TAC, new TAC, old
I
nucleotide?
tested everything.
You tested everything. So what does everything mean? New
I-
water, old water?
Yes. The water was very crucial.
She's joking.
Yeah, but I don't
You
know.
tested TAC, nucleotides?
My Google Docs won't open, so I don't really know what the 2 that work. But we'll find that out eventually.
Well, things work.
It'll work.
Okay, cool.
It'll happen.
So, in class, we've learned so far about mRNA, right? We learned about rRNA. What is rRNA? Ribosomal RNA. It's RNA that's part of the machine that does translation, right? What's another type of RNA that you've learned? tRNA. I call it the taxi RNA. It's the RNA that carries amino acids from the bank to the ribosome to make it protein. Does anyone know of any other RNAs?
sRNA.
What?
sRNA.
sRNA. What is sRNA?
Silencing RNA.
siRNA. That's what you're saying. Okay? MicroRNA. Silencing RNA, microRNA, snRNA. Oh yeah! What does snRNA do? I cannot remember, I just know. We had the SNRPs. What were SNRPs used in?
RNA processing?
RNA processing, splicing, yeah. They're the guide RNAs that help the spliceosome find where to cut, right? Okay, what other RNAs have we heard of? Single-stranded, double-stranded, okay, yeah. So you have most of these we think of as single-stranded RNAs, but yeah, so we get a single-stranded RNA. double-stranded RNA. Okay.
You do positive or negative strand providers.
Okay, positive or negative strand, sense or anti-sense. Okay. These are the RNAs. What does a silencing RNA do?
It binds to certain parts of the transcribed RNA so that it isn't produced by the protein
It binds to certain parts
of the messenger
of a
RNA
messenger RNA,
so that it's not translated.
so the messenger RNA is not translated. So let me explain this fundamental concept to you. So we've got our messenger RNA, right? And you say the microRNA binds to this, like that? You don't know, okay. Let me tell you accurately what happens. So the silencing RNA is complementary to a small sequence in the messenger RNA. And it hybridizes like that. And it shuts down expression, translation of this. Anyone ever hear of that before?
The whole thing or just that part of it?
That's a great question. The whole thing or just that part of it? That's a great question. I'm not going to answer that right now. I'm going to have a few other questions. How many people have heard of silencing RNAs? When I was in graduate school, they were called antisense silencing RNAs. And the mechanism as to how it worked when I was in graduate school was proposed to be that the ribosome, I'm gonna run out of colors of pens here, the ribosome, as you recall, binds to the cap and scans down and starts translating from the initiation codon, right? Remember, we learned that already? What this does is the ribosome is working its way down, and maybe you've got some amino acids started. This thing physically is in the way, so the ribosome can no longer move beyond it. And the effect is, if we had hybridized this thing down here, we would get a whole string of ribosomes all lined up and backed up here, each one with a progressively longer amino acid sequence poking out, because it's translated farther down, right? But eventually, if this was really small, like I've shown here, the ribosome might have enough oomph to nudge the silencing RNA off and go on. So what you would need is this to be a pretty good size. Does that make sense? Okay. Silencing RNA. There is a company, used to be located in Boulder, and the name of the company is Cerna. And it was a company, the mission of the company was to identify how this concept could be applied to treat human disease. For example, let's say you are infected with HIV, human immunodeficiency virus. The idea was this company could manufacture a complementary nucleotide that they could inject into you. That complementary nucleotide would bind to HIV transcripts, shut down expression of HIV proteins, and then you're done with your HIV. How's that? Sounds too easy. Okay. siRNA, microRNA. You said microRNA. What's a microRNA?
It's a small piece of RNA, and I think it has like catalytic properties. Like it'll splice the messenger RNA if it binds to it.
So it is a small RNA, and it binds to messenger RNA, and it splices the messenger RNA.
Mm-hmm. So it degrades it.
It cuts the messenger RNA. So that if it were to come in and somehow have an enzymatic activity that it cuts this guy doesn't have a poly A tail, and so the poly A tail side gets chewed away, and this doesn't have a cap, and so the cap gets this side from the 5' end gets chewed away. Okay. How many people have heard of microRNAs before? Okay. So we're going to read a paper that was published in 96 or 98. 96. was published from the laboratory of Fire and Mello. 2 different laboratories, I think. And they were looking at this antisense type system. These are basically the same thing, it turns out. 2 different ways of saying the same thing. They published their initial paper. If they published it in, I think they published it in 1998. And I think they won the Nobel Prize in 2004. So generally what happens is someone finds something and they spend 30 or 40 years of their career documenting this and building this huge body of literature and showing this amazing thing. And when they're old and really, really old, just before they die, that's when they get the Nobel Prize. So these people had their initial discovery, and 6 years later were awarded the Nobel Prize. I've never in my life ever heard of that. But what they discovered is so profoundly important for expression of for expression of genes in eukaryotes. It was worth the Nobel Prize. It's incredible. So, what we're going to do is we're going to, by the way, I mean you. We are going to read that first paper that they published. And it's really hard to read these things. So, we're going to break down into groups. I think I probably should assign the groups. Should you guys assign the groups? Should I assign the groups? You guys pick your own groups? It's a tie, 3-3. Brilliant. Can you do rock, paper, scissors? I win, I think. It's all riding on
Out
it.
of 3? Like best out of 3 or best out of 1?
Best out of 1. Yeah. Survival of the fittest right here. Okay, so we go 1, 2, and on 3 you pick your name. Ready? Rock always wins. Shit. Pretty good at this game. All right.
So I guess we can.
You guys get to pick. You get to pick your groups. Each group will be assigned 1 paragraph. And you're like, oh, totally piece of cake. My guess is it takes you an hour to read the paragraph and get a good understanding of it. Each paragraph is associated with a figure, a table, a picture, or something. If you get together in a group and you are done with your project in 4 hours, I will be surprised. 1 paragraph. I can sit down and read this and go, oh, yes, 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 yes. I can read the whole thing in an hour. I can be done with it and totally understand it. Well, now that I've read it a couple times, I can do it in 15 minutes and be done with it. Part of the challenge of graduate school, this is my graduate experience, is I went to graduate school, there's no textbook. And the reason is, if you're in grad school, you're at the end of knowledge. The only way you're going to learn is you need to read the paper that came out last month that talks about the new knowledge that was just created. So for me, my experience in graduate school was learning how to read papers. And when I got there, I was like, damn, I can't understand any of this stuff. How the hell am I supposed to read this paper? I have 4 papers to read for my lecture 2 days from now, and each one is going to take me 4 hours to read and understand. I don't think I want to be a scientist. The process of graduate school, the first 2 years of graduate school, was me learning how to read a paper instead of 4 hours to read a paper in 15 minutes and get it. It takes practice. I want you to start on that right now. It doesn't matter if you go into petroleum, or if you go into medicine, or whatever you go into. There will be an area of expertise. You are the cream of the crop of the world. You have to remain experts. You have to remain on top of this. And you have to be able to read up-to-date research and such. So we'll read this. Each group, I would like you to prepare a PowerPoint file, to prepare a 3 to 5 minute presentation which explains the paragraph. I want you to be able to explain, here's the figure. This is the experiment that they actually ran. Here's the data shown in this figure. And it means this. We'll go out, each group will go up in order, and it'll take us 2 days to go through the whole paper. Okay? So, I can't remember how many paragraphs in here. There's a lot of people not in here today, so I'll pick those people and put them into different groups. Orlando and Katie, I don't know how we're going to make this work for you. Maybe you guys make a video. Maybe the two of you are a team and you make a video and you post it on YouTube, and then I show that to the class. It makes the question and answer period kind of difficult afterwards, but so you guys are a group. I'll assign you a paragraph. So why don't we take 2 to 3 minutes right now and break down into groups of 3. If you want to be bold, break down into groups of 2. If you want to get into kind of a, I think if you have 4, there's going to be too many people for your group. So I'm going to give you guys a couple minutes. Great. I'm going to run to the bathroom real quick.
Yeah, that sounds good. I'll be seen with you too. It's just easier to be all
Alright, when you've got a group, and it doesn't, if you sign up for here, it doesn't mean you're getting paragraph 1. I'm going to assign it randomly, alright? When you pick your group, just write your names up here in your group. And just pick randomly picking up. I'm going to randomly assign or the closing paragraph.
So then we come in at 2 to about
Yep. If anyone wants, I'll be around until, actually today I can be around until shortly after 4.
The floppy don't stop.
Take Jacob's head. And it turns out somebody ran a little bit of gel already out there.
American Beagle outfit. Guys,
Somebody had to make this.
powerhouse. Nice up there. Everyone can know. Uh-oh. Sweet.
That's two.
Oh my god. They have their own website. Oh my god. This is like, I first one it was their one. They have dog hats.
So you'll be well practiced. One day better for you.
But you do want to look around, right? Dude, what if they have like a sick, like a saggy hat you just yeah, I'll text you if I want to get one for you.
CAI.
Oh, shop this look for women. Shop this look for women. Oh my god, you can I don't want to 23.
Enjoy it.
Except Aisha, she's like.
Yeah.
See, I know that from a Stevie Wonder song.
I love that song. I don't know that's what American Beagle outfitters have a matching line of clothing now for your dog. Oh my god, that's great. You need to get the dress for your dog and yourself. I like that. And then I'm really stressed out about he's a little bit behind. They painted his toenails.
Wow. Ryan and Kate stepping up to the plate here.
It was him.
They are not even enrolled in the class.
JK! You guys are brave.
And they are signing up here. Good job. Gold star for you today. Remember, you could lose it if you screw up in bio 2 next time.
Okay.
Why would you do it if you're not enrolled in
Wow.
this class? You're just taking it for fun?
They come here for fun. They're weird.
They're taking it for fun. They're taking it because it's so entertaining. It's the best use of their hour, their lunch hour, right?
I just watch like the videos.
Are you gonna talk today? I guess it was
I'm going to tell you today. Yeah, I got to tell you right as soon as possible.
seriously?
I'll post the paper online and I'll have to email it out. Why is the computer so slow? There we go. All over the
Adjust the wires.
It's kind of like that's it.
Are you not here the Monday and Wednesday after you guys?
That doesn't matter.
I am not here the Monday and Wednesday after E-Days, but we will have a guest speaker. Josh Ramey. You guys know Josh?
Yes, I know him.
He teaches Bell Bio 110.
Yeah.
His PhD and area of expertise is basically the same as mine. And he will be coming in the Monday and the Wednesday after because I will be at the beer judging event for the World Beer Cup. Enjoy. You bet I will. It's work. For any, if my boss is watching, he knows it's work. Okay. So, we've talked about gene transcription in very general terms. So Monday, wow, it's taken me 20 minutes to say we're going to do presentations on Monday and Wednesday about microRNA. It's silencing RNAs. So, we learned already about, I think there's 15 paragraphs, so maybe I'll need to jump in and do a couple. Right? So we've done this before, right? What's the name of this thing? Promoter. What's this thing? Plus one or? Start of transcription. Good. What's this? Start codon for translation. Methionine, right? And then so this is the what? What is ORF stand for? Open reading frame. Okay. So we've learned about gene expression. This is what I learned in graduate school. All the other stuff was discovered while I was in graduate school. It's pretty amazing. So when I started graduate school, this is what we knew. We got the RNA polymerase, or a transcription factor that's hanging on RNA polymerase. Binds to the promoter. The function of the promoter is to measure a set distance, and there's plus one. What we learned earlier this semester is if you move the promoter this way, like if we took 20 nucleotides and stuffed it in here so that there were some extra nucleotides in there, what would happen is plus one would shift this way, 20 nucleotides, right? But we would still transcribe off of here, right? What happens if I take my promoter and I turn it around like this? What will happen is the transcription factor will bind here, it'll measure out, RNA polymerase will measure out and will start transcribing this way, right? That's classic gene expression. While I was in graduate school, we learned that whether the DNA is compacted or not determines whether a gene is expressed. A gene that's compacted in DNA, RNA polymerase can't find the promoter because the promoter is buried in this big snarl of DNA. So they learned about the histone code and they learned about DNA methylation. It turns out there was actually a guy on my thesis committee who discovered a new feature of DNA that we didn't know exists. These are called enhancers. We'll talk about enhancers today. So, why, first of all, why would we want some gene to be expressed and some genes not expressed? And why would we want to control gene expression? So this is what's called a Western blot. Has anyone ever heard of Western blotting? A couple people? Does anyone want to dare an explanation about how a Western blot works?
So Western, Eastern, one's DNA, one's RNA, right? Is that how
There's
it
no
works?
Eastern.
Or is it? Oh, I think it's Southern and Northern.
There's a northern blot. So the first blot that became available was a southern blot. And I can't remember, Dr. Southern, that's his name, came up with it. And it was in order to identify, it was a way of identifying RNAs on a piece of paper. So someone said, well, if that's how you identify, if a southern blot, if you blot by the technique of southern and you identify RNA, if you're to do the same thing but identify DNA on a piece of paper, then we'll call that a northern blot. And someone said, a western blot will be what? Protein. That's right. And there's no eastern blot. Because that's it. It's RNA, DNA, protein. Huh?
We're just done forever. There's no more discovery.
Yeah, there's no more discoveries. That's it. So everyone's excused to see you guys after eating. I'm just kidding. Do you know what happens in a western blot, how it works?
Is it based on charge? Is like the size? They have more negative charge on it or something? I can't remember.
Yeah, okay.
I kind of remember. Isn't it like fragmented DNA is put through like gel,
Okay.
and then it's like transferred to like some type of like sheet,
Yep, it's called nitrocellulose.
that kind of sheet, and then from there you like use a tag, like a can't remember what kind,
Right.
and then that like identifies the specific DNA that you're looking for.
So let's do a northern blot with DNA. Okay. So we take an agarose gel. There are wells up here. We load the DNA in. We apply a charge. The DNA separates. So what's different? What's different about this DNA compared to this DNA? Size. This one's smaller than this one, right? So let's take the human genome and digest it with this enzyme called EcoR1. So the DNA genome is 3.2 billion nucleotides. How many DNA fragments do we get if we cut the human genome with EcoR1? Who says more than 10? Who says more than 1,000? Okay? You're going to get, if you ran that DNA out, you're going to get so many different sized DNA bands that it's going to look like this. That's real helpful. Okay? What if we want to know where the gene for, someone name a gene. What's your favorite gene?
TMEM106A.
TMEM106A. We want to know which of these fragments has TMEM106A. Okay? Turns out, I'm going to actually take a piece of paper. It's a special piece of paper made out of nitrocellulose. Don't worry about that. I can put the piece of paper on here, and then I can actually apply a charge down here, a negative charge down here, and a positive charge up here. And the DNA says I want to get the hell away from the negative charge, and so it runs this way. And the reason we use nitrocellulose is nitrocellulose is sticky for DNA. So that when the DNA comes out of the gel, it runs into the nitrocellulose paper and gets stuck on the paper. So now, I have a piece of paper in which all this DNA is laid out on it like this. Which one of these bands has TMEM106A? What I can do is I can call IDT and order a primer like you already have, and I can tell IDT I want my primer to be radioactive, and I want that sequence to be a sequence that will bind to TMEM106A. I take that primer, put it in solution, and wash that solution over this paper. And now, any place that the sequence TMEM106A is located, the radioactivity will bind to that, right? And then I just wash all the excess off, and I can, for simplicity's sake, I can take a Geiger pen and go, quiet, 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 and then there's a really hot spot on the piece of paper. That's the size of TMEM106A. Does that make sense? In theory, in reality, what we do is we take a piece of photographic film and lay that on top. As radioactivity decays, it releases photons of light, and you develop the film, and you can see where exactly which band has the DNA. Does that make sense? A Western blot is that same thing. you have a gel that you load proteins into. So you can take cells, you can load the cells into the well, and you turn on the charge, and the proteins separate out by size. So small proteins are down here, and all of the cells, all of the proteins from the cell will then be separated out. You transfer the proteins onto a piece of paper, you can make a chemical that binds to your protein specifically. There is a protein, which I'm sure everybody here has heard of, called actin. What does actin do? It's in muscles, right? It's a motor. So it turns out every cell in your body makes actin, and every cell expresses about the same amount of actin. Muscle cells have more, but that's fine. So that's called what we call a housekeeping gene. If you went and took out brain tissue for me, please don't, took out brain tissue for me, and you put those cells in here and ran them out, and then you went into my liver and took those cells and put them over here and ran them out, the amount of actin per cell in this lane in the brain and the amount of actin per cell in the liver is about the same. It doesn't change. And we've actually run a Western blot, and we see the amount of actin in this Western blot is the same. And it doesn't matter. And I guess what they've done is, over time, in this well, what they've done is they look for, it looks like cyclin D3. I can't really read that very well. And it turns out they're watching as a cell has divided, how much of the cyclin D3 protein is present in the cell. And immediately as the cell divided, there's a lot of it, but then the amount of this protein decreases over time, until essentially there's none inside the cell. So cells have a way of turning genes on, and they have a way of turning things off. Yeah?
How do you run a gel when you have like neutral or positive charged proteins? It's like amino acids.
You are asking a great question. You're way beyond the scope of this course. Let me answer that. So his question is, what's the charge of, let's do this. So generally you say I've got a negative charge here, and I have the positive electrode down here, right? So DNA will separate out by size, right? If I take proteins and load proteins in here and run them, what's the charge on a histone? Huh? Positive. That's why it binds to DNA. Is that protein going to come and separate on this gel down here? No, it's going to run this way. Whoops. So that was a great question. How can you separate by size? If positive, you could have a huge protein. It's positively charged. It's going to go this way. And what happens is there's a special chemical, and we put the proteins in there, we add a special chemical on it, in there also called SDS. You know what SDS is? Does anyone ever work with SDS? Yeah? Sodium didosyl sulfate. Probably today, every one of you has used SDS. You know what it does?
It's a detergent.
It's a detergent. It's the bubble stuff in your shampoo and soap. That stuff binds to proteins and covers the proteins with negative charges, so that every protein is very, very negatively charged. So now every protein runs this way and they separate by size. That's way beyond the scope of this course, but it's awesome for you to ask. Yeah?
Are they first separated by charge and then run it through an SDS gel?
There are multiple ways to do this. You can do it, yeah, you can do it by charge, there's all kinds. By hydrophobicity, it's not going to do it. Okay. Now, some genes are on and some genes are off. This is, believe it or not, a cross-section of brain. Oh, let's say it's a mouse brain. Let's not say it's a human brain. They're looking at expression of some particular gene, and they couldn't find it expressed anywhere. And in the old days, like when I was in graduate school, what they would do is they'd say, okay, I found this new gene in the mouse genome. I don't know what it does. And what they do is they would take a mouse that had died of natural causes. Okay? A mouse in the lab that had died of natural causes. And they would dissect the mouse, they'd take the liver, and they would grind the liver up, and they'd run all the proteins out. And they'd say, is the protein for my gene found in liver? And they run a Western blot, and there's nothing shows up. So it's not expressed in the liver. And they'd say, okay, here's the mouse brain. And we'll mush up the mouse brain, and put some mouse brain here, and run it out, all the proteins out. Oh, we find my protein expressed in brain. Okay, so it must be that all neurons in the brain express this gene. Well, it turns out what they've done here is they've actually done a cross-section of the brain, and they have looked, they basically run this Western blot protocol on the brain tissue itself. And that protein is expressed in these dark spots, and it's not expressed any other place. Oh, it looks like a couple neurons here have spotty expression of it. But generally, this gene is expressed only in certain parts of the brain. It's amazing, isn't it? How do the cells control this? We'll talk about that in this next couple lectures. So, any chemical engineers in this class? Right. Any chemistry majors in this class? Who is not a chemie or a chemistry major? A couple of you. So, mechanical engineering? What is it, Travis?
Physics.
Physics? Okay. Huh? Just weird. Okay. So, we've got our central dogma of biology, right? DNA, pre-RNA, mRNA, protein. To a chemical engineer, does that look like a continual feed process? Yes or no? Do you guys know what a continual feed process is?
Yeah.
Is this how you would diagram a continual feed process? No? Can I get some comments on this?
Yes.
Yes? To a chemistry major, does this look like a chemical pathway?
Yes. Yep.
Yes. Can you model this with the beautiful Michaelis-Menten equation? Probably. Wouldn't that be unpleasant? We're not going to do it. I promise we're not going to do it. Right? Can someone explain to me, if I apply the term steady state, real basic stuff here, what does it mean when I say steady state to this?
Well, the overall value or concentration or whatever isn't changing with respect to time.
So the concentration, if I've got 10 molecules per minute being made in this process, I'm losing 10. So this is 10 molecules per minute going in. So I've got 10 molecules per minute going out. Right? Such that the number here stays constant over time. Right? That's how you apply steady state to this? And you guys can mathematically model all that? I'm going to trust you on it. There was a time that I could, and I've forgotten. I'd have to go back and relearn how to do that. You can model all of this, right? So it turns out a cell can change whether a gene is being expressed or not by changing how much is going in. So the cell could say, oh, I'm going to increase this to 100 per minute, which means I now have this swell of material coming in. We're not at steady state anymore. But ultimately what will happen is this number will become larger. And now I'm expressing it. What's the other way of increasing the amount of this, and I'm not tinkering with the amount coming in?
By stopping what's leaving.
Yeah, shut this down so that only 1 a minute is being degraded and eliminated. Now we're not at steady state anymore, and slowly this will accumulate until we reach a new steady state, right? Because slowly as you accumulate this will go to 2 a minute eventually, but you get this huge swell here. Does that make sense? Does this make sense to the chemies? You learned about this like in excruciating detail. How about physics? You learned about this type of stuff?
I actually just switched out of chemistry.
You just switched out of chemistry, so you definitely know this stuff. Yeah. This is why you left.
Yeah, exactly.
In a biochemical pathway. You guys have modeled this in biochemical pathways, haven't you? In chemistry majors? This is not new anyway. Okay, great. So we can use these very fundamental concepts to illustrate how we can regulate gene expression. So it turns out there are a couple of different ways that you can exit from the system. We're going to learn about microRNAs. MicroRNAs are a way of taking this and you degrade this into nucleotides. Sorry. Nucleotides. Processes degrade. Right? This is process. And this This is translate. Am I doing this right? This is transcribe. Okay, so if we're doing 10 a minute, and now suddenly we start degrading 5 a minute, we're going to have less accumulation of this product right here, right? It turns out with proteins, you can degrade proteins. This is a standard way of getting rid of them. You degrade them into amino acids, right? But you can actually take proteins out of this process, or out of this state. So let's say that this is some enzyme. Let's say this is hexokinase. As you of course know, hexokinase equals glucose to glucose 6-phosphate, the first step in glycolysis. What if, let's say hexokinase, let's say amazingly hexokinase looks like a little Pac-Man here. And that this is the active site of the enzyme. So glucose comes in, binds, and you put a phosphate group on it. What if I were to take something and stick it in here, like, I don't know, let's say a methyl group. Now glucose physically can't fit into this spot. This is no longer hexokinase. This is methyl hexokinase, right? We put a methyl group on it. Now usually you don't put methyl groups on proteins. So typically what you'd want to put on here is kind of like a phosphate group. So we could actually decrease the concentration of protein by moving it to a phosphorylated state. Right? And the beauty of this is, if suddenly we need a burst of energy, what we can do is we just take this phosphate off of here and move it back to a protein, and it's active again, and now we can process, start a whole bunch of glucose running through glycolysis again, right? So I guess the arrows go this way. It's reversible. Do that, right? So there are many different ways, many fine tinkering controls of accomplishing this. And I kind of illustrate that here in the next couple slides. This is what I learned when I started graduate school. If a cell needs more hexokinase, what I learned is there's 2 ways to accomplish that. RNA polymerase binds to the DNA and you make more pre-RNA. Now that you have more pre-RNA, more of that gets processed into messenger RNA, more of that gets processed into protein. We had no idea there were ways of controlling this. We figured there were probably ways of controlling this, and we knew about this. But in general, if you were going to increase the amount of protein, you increased transcription first. Right? That's classic gene expression. And while I was in graduate school, we learned about this microRNA, these silencing RNAs. It's a way, even if you have a massive amount of this stuff going through here, it's a system for taking the messenger RNA before it can be translated and destroying it, so that almost no protein gets made. Why would you want to burn all the energy to make this huge transcript that you then process into a final product, and you don't make any protein off it, you destroy before you ever translate off it? Why would you want to do that? It does sound very strange. There are genes that definitely, this is steady state. That a cell and normally is in this situation. You got an answer? You're fidgeting in your seat.
Well, to me it would seem that it would be an extra process in cell signaling, that if it needed that protein right away, then it would just, instead of degrading the mRNA, it would translate it directly.
Yeah. So
there's always a new incoming flux of the mRNA.
Right.
That never gets touched.
Right. Yes. Good. So let me rephrase this. I'm going to put both of your things together. So we have a cell that's just sitting there. It gets hit with some environmental signal, and that cell says, "Oh, we've got a whole bunch of lactose. We need to tap into this lactose now." So a whole bunch of transcription factors get activated. RNA polymerase binds to the DNA, measures out, we transcribe, takes half an hour, maybe an hour, you get a whole bunch of transcripts made, now half an hour to an hour, half an hour to process those into messenger RNA, and then half an hour to process those. It's now been an hour, hour and a half, maybe 2 hours. We now have a whole bunch of protein. We start processing the lactose. There's no lactose around anymore, it washed away. Whoops. However, if the cell is always making the genes to process lactose, but they're being destroyed, when the cell gets hit with lactose, all it does is say, stop degrading, allow the messenger RNA to get translated. Now, 20 minutes later, we've got a massive amount of protein to process the lactose, and we're still in this environment. We harvest all that energy. Make sense? So there are genes that are so important to get an immediate response that it's worth burning all the energy and wasting it in integrated RNA for the one moment, that one time you need to have it. There are genes like that out there. So we can modify these things as other ways. So I want to add, and it's not on here, I want you guys to tell me, how do I modify this expanded central dogma of biology to account for DNA packaging? You guys have been hugging it over here this time. How do I modify this so that I'm adding DNA packaging into this? Anyone guess?
It's kind of like with the protein, only it's modifications to the DNA.
So it's kind of like modifying the protein, modifying the DNA.
There was something on the down arrow kind of deal somewhere.
So what is this DNA then? Open DNA, unpackaged.
Ready to go.
And this is compacted DNA.
Heterochromatin?
Huh?
Heterochromatin?
Heterochromatin, yeah. This is euchromatin. This is heterochromatin, yeah. Or packaged. Okay, good. Thank you. Someone else had their hand raised, they were going to say something. Are you going to say something?
Yeah.
All right. This is pretty much the central dogma that I want you guys to learn. When you walk out of this class, I want you to be able to draw this. That's one of my objectives that I write to myself, that I want every student to be able to draw this and understand this. In General Biology 1, you probably learned this. And now you move to genetics, you move to an upper level class, we need to add some more features onto this. That's it. So if you don't have this written down, write it, take a picture, see if you have a pen. Questions on that? So I think on exam 1 I said draw the expanded central dogma of biology using 4 terms and 3 words. Or 3 arrows and 4 terms or something like that. Now, this is what if I ask to draw the central dogma of biology on an exam, this is what I'm asking to do. And if you take graduate level, it gets more complicated than that. Do you need me to say these words? You can't read these words?
Just one word, a keyword.
This says heterochromatin
No, not
or
all.
packaged DNA.
This
is open or unpackaged DNA. This is euchromatin.
not any of those. After the pre-RNA, just in between that process.
Processing.
Process, okay.
RNA processing, you call it splicing, capping, polydenylation. It's 3 steps, right? Oh my God. I can't believe I wasted my whole hour here. I didn't even get into my lecture.
I know.
All right, finish this up.
Well, that wasn't meant to sound sarcastic. That was genuine. But